Today we are going to delve into an exciting and integral part of bioinformatics, proteomics. Let's unpack what this is all about and why it's so important in biological sciences. Firstly, what exactly is proteomics? So imagine you are in a vast library. Instead of books, it's filled with proteins, the essential molecules in our bodies. Proteomics is the study of these proteins, specifically their structures, functions, and interactions in living organisms. It's like being a librarian, cataloging and understanding every book in this vast library. Why is proteomics crucial? It plays a key role in understanding the intricate dance of cellular functions. Proteins are involved in virtually every process within our cells, from providing structural support to catalyzing chemical reactions. By studying proteins, we gain insights into how cells function, how they communicate, and how they respond to changes in the environment. Proteomics doesn't just stop at studying individual proteins, it extends to understanding how the proteins interact with each other and with other molecules in the cell. This comprehensive view helps us decipher complex biological processes and diseases at a molecular level. As we delve deeper into proteomics, let's understand its diverse approaches. Each type of proteomic study gives us a unique perspective on proteins and their roles in the body. First is expression proteomics. So imagine a bustling city where each inhabitant has a specific role. Now replace the city with a cell and the inhabitants with proteins. Expression proteomics is like taking a snapshot of this bustling city to see who's there and what they are doing. It identifies which proteins are present in a cell at any given moment and in what quantities. This helps us understand how the cells respond to various conditions like stress, medication, or disease. Next, we explore functional proteomics. So if expression proteomics tells us who is in the cell, functional proteomics tells us what these proteins are doing and how they are interacting. This approach involves mapping out the interactions and the roles of the proteins in cellular processes. It's like observing the interactions and the duties of the city's inhabitants. This is key for understanding the complex mechanisms like how a cell decides to divide or how it responds to a drug. Lastly, we have structural proteomics. It's also about understanding the 3D shape of the proteins. Why does shape matter? Because the structure of a protein determines how it functions and interacts with other molecules, including potential drugs. It's akin to understanding how the design of a tool defines its purpose. Structural proteomics gives us insights into the physical form of proteins, helping in drug design and understanding the diseases at a molecular level. Together with these proteomic approaches, expression, functional, and structural provide a comprehensive picture of the proteome, and they enable us to decode the complex language of proteins, leading to the breakthroughs in our understanding of health and disease. Let's now focus on the basics of the protein structure. Understanding this is crucial as the structure of a protein is intimately linked to its function in the body. First, we have the primary structure. Imagine a protein as a string of beads where each bead represents an amino acid. The sequence of these amino acids in the chain is the primary structure. This sequence, determined by the corresponding gene, dictates how the protein will eventually fold and function. Next is the secondary structure. This level is about how the amino acid chain starts to fold. There are two common patterns, alpha helices like spirals and beta pleated sheets like folds. These structures are formed by hydrogen bonds between amino acids in the chain and give the protein its initial shape. Moving to the tertiary structure. This is where the protein takes on a complex 3D shape. It's the result of the chain folding and twisting in a specific way. driven by interactions between the amino acids, like hydrophobic interactions, ionic bonds, and disulfide bridges. This level of structure is crucial as it determines the protein's functional shape. Lastly, we have the quaternary structure. Not all the proteins have this level, but for those that do, it involves the combination of several protein subunits. These subunits come together to form a functional protein complex like the hemoglobin molecule in our blood. Now, to sum up, each level of protein structure is vital in determining the protein's final shape and thus its function. A change in structure at any level can significantly affect a protein's functionality, which is why understanding the protein structure is key in fields like drug design and disease treatment. Having understood the basics of the protein structure, let's now see how scientists predict these structures. Protein structure prediction is a vital area in bioinformatics, crucial for understanding biological functions and developing new drugs. Why is predicting protein structures so important? The 3D shape of a protein is key to how it works. However, determining the structure experimentally through methods like X-ray crystallography or NMR spectroscopy can be time-consuming and challenging. Computational prediction allows us to rapidly hypothesize the structure of a protein from its amino acid sequence, providing invaluable insights, especially for proteins that are difficult to study experimentally. One common method is homology modeling. If we know the structure of a similar protein, we can predict the structure of a new protein based on this template. It's like using the blueprint of one house to estimate the design of another similar house. This method works well when the new protein shares a significant sequence similarity with the known protein. Then there are ab initio methods, which means from the beginning. These methods predict the protein structures from scratch, based solely on the amino acid sequence without relying on a template. It's like building a house from the ground up using only the list of materials. This is challenging and typically used for small proteins. Recently, machine learning approaches like AlphaFold have revolutionized protein structure prediction. AlphaFold uses deep learning to predict the protein structures with remarkable accuracy. It's like having an AI architect who can design a detailed building plan based on past architectural knowledge and patterns. Predicting the protein structures helps in drug design, understanding disease mechanisms, and even designing novel proteins. For instance, accurate structure prediction was crucial in quickly developing COVID-19 treatments and vaccines. Now let's explore some of the essential tools and databases in proteomics. These resources are crucial for analyzing the proteins and understanding their functions in detail. First, let's talk about the tools commonly used in proteomics. Imagine being a detective. To solve a case, you need specific tools. In proteomics, two such tools are mass spectrometry and 2D gel electrophoresis. Mass spectrometry is like a sophisticated scale. It measures the mass of protein fragments, helping us identify and quantify proteins in a sample. It's incredibly precise, capable of distinguishing even minute differences in protein masses. This makes it essential for analyzing the complex protein mixtures and post-translational modifications. Then there's 2D gel electrophoresis. Think of it as a way to separate a crowd into groups based on height and weight. In this case, proteins are separated based on their isoelectric point, or the charge I should say, and molecular weight. This method allows us to visualize the protein composition of a sample, often as a first step before more detailed analysis. Now on to the databases. Two key databases in proteomics are the PDB, that is Protein Data Bank, and UniProt. The PDB is like a 3D gallery of protein structures providing detailed structural information about proteins and nucleic acids. It's invaluable for researchers studying protein structure and function. UniProt, on the other hand, is like an encyclopedia of proteins. It offers a comprehensive resource of protein sequences and functional information collected from various sources. It's a go-to database for anyone looking to study protein sequences and their biological roles. Let's take a closer look at UniProt, one of the most comprehensive and widely used protein databases in the field of proteomics. UniProt, that is the short for Universal Protein Resource, is a central hub for protein information. It's like an encyclopedia dedicated entirely to proteins. The database is designed to provide detailed information about the functions, structures, and the roles of proteins across various species. UniProt is comprised of several key components. The first one is UniProt Knowledge Base or UniProt KB. This is the heart of UniProt. It has two sections: SwissProt, which contains manually annotated records with information reviewed by experts, and Tremble, which includes automatically annotated records waiting for review. This setup ensures that UniProt contains both highly accurate, curated data and a comprehensive collection of protein sequences. Next is UniRef, UniProt Reference Clusters. These are the databases that provide clustered set of sequences from UniProt KB. They help in speeding up sequence similarity searches by grouping the sequences that are closely related. Next is UniPark, UniProt Archive. The most comprehensive collection of protein sequences. UniPark stores sequences from various sources, ensuring that each sequence is only represented once. UniProt offers several features. Comprehensive protein information, from basic sequence data to detailed annotations about function, structure, and interactions. It provides links to other databases and resources, providing a holistic view of protein data. UniProt provides tools for sequence analysis, protein domain identification, and functional annotation. It's invaluable for researchers in bioinformatics and related fields. It's used for everything from basic research in understanding protein functions to applied research in drug discovery and disease diagnosis. The depth and breadth of its data makes it a primary resource for scientists worldwide. Now, let's explore how proteomics is making significant contributions to research and medicine. The advanced understanding of proteins is not just academic, it has real, tangible impacts on health and disease treatment. First, let's consider drug discovery. Proteomics is like a detective working to unravel the mystery of diseases at the molecular level. By identifying and understanding the proteins involved in diseases, researchers can develop drugs that target these proteins more effectively. For instance, in cancer treatment, drugs can be designed to specifically target the proteins that are overactive in cancer cells, leaving healthy cells largely unaffected. Next is disease diagnosis. Proteomics can identify unique proteins or the patterns of proteins in diseases known as biomarkers. These biomarkers can be used for early diagnosis or to monitor the progress of a disease. For example, specific protein markers in the blood can indicate the early stages of heart disease or certain types of cancer. Then there's personalized medicine. Every individual's protein makeup is slightly different, and these differences can affect how we respond to drugs. Proteomics allows for a personalized approach to treatment, where therapies are tailored based on an individual's proteome, leading to more effective and safer treatments. As a real-world example, let's look at biomarker discovery. In conditions like Alzheimer's disease, certain proteins become misfolded and accumulate in the brain. Proteomic studies have identified these proteins, which can now be targeted for early diagnosis and treatment development, providing new hope in combating this disease. In a sense, proteomics plays a crucial role in advancing our understanding of diseases, developing new therapeutic strategies, and personalizing medical treatments. It's a field that bridges the gap between basic biological research and tangible medical benefits. As we look towards the future of proteomics, it's important to recognize both the challenges we face today and the exciting possibilities on the horizon. Proteomics, despite its advancements, faces significant challenges. One major hurdle is the complexity of the proteome. Unlike the genome, which is relatively static, the proteome is dynamic and changes in response to various factors. This complexity makes it difficult to fully understand the functions and the interactions of proteins. Another challenge lies in the sensitivity and the accuracy of the current technologies. Detecting low abundance proteins, which can be crucial for understanding diseases, remains a difficult task. Also, interpreting the vast amounts of data generated by proteomic studies requires sophisticated computational tools and algorithms. Looking ahead, the future of proteomics is bright and full of potential. One exciting trend is the integration of artificial intelligence and machine learning. These technologies promise to enhance our ability to analyze and interpret complex proteomic data, making discoveries faster and more accurate. Another emerging trend is the development of more sensitive and precise tools for protein analysis. These advancements will enable us to detect and study proteins that are currently difficult to analyze, deepening our understanding of biological processes and diseases. As we conclude today's lecture on proteomics and protein structure prediction, let's recap the key points and look at resources for further exploration. We started with an introduction to proteomics, understanding its vital role in deciphering the complex world of proteins. We then delved into the different types of proteomic studies: expression, functional, and structural, each providing unique insights into protein roles and interactions. We explored the fascinating realm of protein structure from primary to quaternary levels and the importance of these structures in determining protein function. We discussed the cutting-edge method of protein structure prediction, including homology modeling, ab initio methods, and machine learning approaches like AlphaFold. We also touched upon crucial tools and databases in proteomics such as mass spectrometry, 2D gel electrophoresis, the Protein Data Bank, and UniProt. Lastly, we covered the applications of proteomics in research and medicine, especially in drug discovery, disease diagnosis, and personalized medicine, and concluded with the current challenges and exciting future directions in this field.